Hi everyone, so in this video we're going to be looking specifically at innate immunity. So in our last video we looked at there being three different strands of immunity, the first, the second and the third line defenses. So we're going to be looking at the innate system specifically, which is the first and the second line defenses. So we're going to be looking at the first line of defenses first. So these are all the barriers, the very first barriers that we have in place to protect us from pathogens and disease. So we are going to very quickly look at plants. We're only going to look at plants for the first line defenses. Plants don't really have second or third line immunity. They haven't really evolved anything like that. So they strictly rely on their first line defenses because there are pathogens that actually attack plants as well. So the first line defenses in plants are mostly physical or chemical defenses. And they're particularly important because they cannot move to protect themselves. So if a pathogen comes to, attack a plant, there's not really much they can do, so they really have to rely on these defenses. So in the diagram here, I've got the physical in red and chemical in green. So some of the physical barriers that plants may have are thorns, thick bark, waxy cuticles on leaves, anything that can prevent the entry of a pathogen into the plant. And then you can have other things like chemicals, like defensins and phenols that can prevent further attacks. They can be toxic, any of that sort of stuff, a range of chemicals, but that's really all you need to know in terms of plants. The rest of this course is going to be strictly related to animals. So in animals, like human beings, we also have physical and chemical barriers, but we also have microbiological ones as well. So that pretty much relates to our gut microbiome, gut flora, all of that sort of stuff. So when we're talking about our first line defenses, we've got physical barriers like our skin and our mucus secretions, and then we've got chemical barriers, things like sweat and stomach acid and lysosomes, anything that can slow down or stop the entry of pathogens inside of our body. And that's really all you need to know about those first line defenses. All you need to be able to do is identify a couple of them, maybe one example of each of the categories, and explain how it could prevent a pathogen from gaining access to our bodies. The rest of this video is going to be about the second line defenses, and I'm about to throw a lot of cell types and chemicals and things at you. There is a lot. I'd probably recommend doing a glossary for 7C because it's not hard, but there's a lot of new terms that you need to wrap your head around. So the second line defenses, they're still the innate immune system, so they're still natural, we're still born with it, they're still very non-specific, but they do work internally to prevent the spread of infection. So if a pathogen does breach our first line defenses, it gets through our skin, gets through our mucus, whatever, this is going to be the next line that's going to be sitting there and it's going to be waiting to further try to destruct that pathogen. So these cells are called either leukocytes or white blood cells. You've probably heard white blood cells before, but they can also be called leukocytes as well. So this little diagram here is just a little snapshot into all the different kinds we're going to be looking at, but I'm going to go through them all one by one, and they all play a major role in protecting our bodies from pathogens. So the first category of innate immune cell that we're going to be looking at are phagocytes. So phagocytes is like a broad term, and we're going to look at three specific types of phagocyte as well. So what a phagocyte is, it performs phagocytosis. Phagocytosis is basically when they consume and destroy foreign material. So phagocytes might completely engulf a bacteria and consume and destroy it. So that's how they work. They have lots and lots of lysosomes inside of them, so hopefully you remember that's an organelle that's involved in just like enzymes that can destroy like waste and things like that. So phagocytes have lots of those and they destroy pathogens. So the three main ones that we need to know are neutrophils, macrophages, and dendritic cells. So neutrophils are the most common type of phagocyte and probably the most common type of immune cell that you're going to have inside of your body. So they obviously engage in phagocytosis, and they also release things called cytokines. So cytokines is going to come up a lot because they're the key communication molecule of the immune system. So basically any time that any immune component wants to talk to another immune component, they're going to release cytokines. And neutrophils do this a lot because they're normally one of the first ones on the scene. So they're going to release cytokines that are going to attract other immune cells to whatever the affected area is. We've also got macrophages. So macrophages, again, also engage in phagocytosis, but they're also something called an antigen-presenting cell. So remember when we were learning about MHC markers, there was class 1 and there was class 2. So I said that class 2 was only on specific immune cells, and macrophages are one of those specific immune cells. So the purpose of an MHC class 2 molecule is to be able to present foreign antigens on their surface. So when a macrophage engulfs something, so we've got a little macrophage there, and he's going to engulf this little bacteria here, what he does is that when he engulfs that bacteria, he's going to recognize the antigen that's on that bacteria, and he's going to wear it like a little hat on his MHC class 2 marker. So that's what MHC class 2 is for. It's to present antigens on the surface. And why is that important? Well, if they're going to present the antigen on their surface, that means that other immune cells can come over and they can recognize that antigen and they know that there's something foreign in the area. So macrophages really take one for the team and they, present those antigens for us so other immune cells can see that something is wrong. Dendritic cells are really, really similar. So they engage in phagocytosis and are also an antigen-presenting cell. So again, these dendritic cells also have an MHC class 2 marker as well. Dendritic cells are probably the main cell though. This is the difference between the two. Macrophages will, present foreign antigens to the cells around it. What dendritic cells do is they present foreign antigens and they go to a lymph node and they communicate with the adaptive immune system. And we're going to learn a little bit about that in our next video. But they're the ones that will trigger that adaptive third line response. So if things aren't going well, if we're not, destroying the pathogen, they are the ones that are going to go and wear their little antigen hat and they're going to go to a lymph node and present that to an adaptive immune cell so that it knows that it needs to start activating. So there are our phagocytes. Then we've got our natural killer cells. So natural killer cells are these large granulated cells and they destroy virally infected cells. So whereas phagocytes are probably more specific to extracellular pathogens, so they can go and engulf things that are floating around, natural killer cells specialize in intracellular pathogens. So they will recognize a cell that is infected with a virus and then they're going to go to that cell and they're going to basically make that cell die. So they're going to induce apoptosis in that cell, and they're also going to tell other cells around it that there is a virus on the loose. So they do this in two different ways. So they work in two different ways. So what happens is that these NK cells, these natural killer cells, they have two receptors. So they have a inhibitory receptor and they have an activation receptor. So if the killer inhibitory receptor binds to an MHC class 1 molecule, it determines whether it's self or non-self. So that's the first step. So first the NK cell needs to recognize it as self or as non-self. So you'll see in this example here where there's no cell death occurring because the NK cell obviously needs to be accurate in whether it's infected or not. So the NK cell is going to come over with its inhibitory receptor and it's going to bind to the MHC class 1 of that cell. So because it's bound to the MHC class 1, it can recognize it as self and therefore it allows the cell to survive. All right, pretty straightforward. So that's how it makes sure it doesn't go and kill every single cell that's around. The important one though is the killer activation receptor. So this is when there is insufficient binding of our inhibitory receptor to MHC class 1 molecules. So when a virus infects a cell, there's going to be certain molecules that appear on the cell surface. So when a cell is under stress, it's going to present different molecules on its surface to try and signal to other cells that there's something wrong. So what's going to happen is the MHC class 1 is going to be unbindable, so the molecules on the surface are going to change, and that means that killer activation receptor is going to bind to those different molecules and is therefore going to initiate cell death. So that does a couple of things. So one, it makes sure that NK cells don't go around killing everything that's healthy. So that's why it's got the inhibitory receptor. But then it also can identify cells that are infected with the virus. It can induce apoptosis or death in those cells. And it can also act as a stress molecule or a stress signal to cells around it so that so the cells around it know that there is a virus and they can change their chemistry accordingly. So natural killer cells are really important in the destruction of intracellular viruses. We've then got our mast cells and our eosinophils. So these ones are quite specific to two different scenarios. So our mast cells they reside in connective tissues. So our mast cells are normally going to be the first one on the scene. So if you step on a splinter or whatever and bacteria gets introduced into your skin, the mast cells are normally going to be the first guys on that scene. So they're going to be the first ones to detect an injury, they're going to be the first ones to detect an allergen, and what they do is they degranulate and release histamine. So you probably heard the word histamine in the context of antihistamine, like you take to prevent allergies. That antihistamine is trying to reduce the effects of these guys here because their first initial response, because they're non-specific, is they will release histamine to any non-self thing that enters the body. And histamine is what causes those really awful like cold, flu, allergy-like symptoms. It's not actually the pathogen itself. So mast cells are the culprit for like sniffly, watery eyes, all of that sort of stuff. So that histamine is a really good marker for other cells to come over and help out. We then have our eosinophils. So eosinophils are really, really, really specific to large pathogens. So these are granulated cells and they have toxins inside. So when they target a pathogen that's too large to be phagocytosed, so if a phagocyte comes over and he's like, nah, this is too big, so it's things like worms and parasites and like quite large pathogens, this is when the eosinophils are going to come, they're going to degranulate and they're going to release toxins and they make contact with the pathogen and then they kill it. So eosinophils are really specific to extracellular large pathogens. We then also have a variety of non-cellular components. So all of the things we've talked about so far have been cells, they've been white blood cells. We also have a range of things that are non-cellular that also work in the second line of defense. So we have these molecules called interferons. So interferons, what they are, they're a type of cytokine. So remember the cytokines are the key communication molecule that are released by a virally infected cell. So when a cell, because they're very humble and selfless, when a cell is infected with a virus, what it does is it releases a bunch of interferons. These interferons are going to interact with neighboring cells and they're going to cause changes that make them less susceptible to infection. So the infected cell is, doing a solid to all of its neighbors, all of its friends, telling them, hey, there's a virus over here. And then the cells around them can act accordingly and change, their chemical physical makeup to prevent the spread of infection. So interferons are really cool. We then have fever, which you're probably very familiar with. This is an increase in body temperature. The reason that we do this is because, obviously bacteria are using, certain proteins and all that sort of stuff. We know that they're very sensitive to temperature. So a rise in body temperature not only kills pathogens and makes them not be able to use their proteins, toxins, whatever, but high temperatures can also help activate and enhance the production of certain proteins in us and enzymes in us to also help aid to fight the infection. So fever, a rise in temp is really, really important. The last one we have here is the complement. So complement proteins they're like a gang that kind of patrols around your bloodstream and it sits there and it waits for this sort of stuff. It loves it. So complement proteins is a big group of proteins. There's about 20 of them and they just patrol around the blood as a little group and they wait and wait and wait for pathogens to come. So when a pathogen does come, these proteins start reacting with each other and cause something called a complement cascade. So the complement cascade can have a variety of different functions and they're described in this table here. So when the complement detect a pathogen and they undergo this complement cascade, they can do three things. They can opsonize, so they can do opsonization, which is basically when they stick onto the surface of a pathogen and it makes it easier for them to detect. So for instance, we've got our bacteria here and we've got all of our complement proteins that are stuck on the outside. So what it's going to do is it's going to make them larger, it's going to make them more recognizable as foreign, and all of the other white blood cells can come and attack it. They can do something called chemotaxis. So chemotaxis is when the complement proteins gather near a pathogen. So you can see over here, so they've gathered near this pathogen, which is basically marking it as, weird. So these complements are like, hey guys, look at this pathogen, and they try and draw attention to it, and that means that the phagocytes and all the other white blood cells can recognize it and come and attack it. The last thing that it can do is lysis. So what can happen is the complement proteins can join together on the surface of a pathogen, and they can create something called a membrane attack complex, which is shortened to a MAC. So this MAC creates pores in the membrane of the pathogen, and it basically causes lysis of the pathogen. So a little bit different to apoptosis, a little bit different to necrosis, but it's lysis. So this is the membrane attack complex here. So this is all the proteins joining up, and what it does is it pokes holes into the pathogen and basically makes it explode and burst, which is pretty brutal, but good for us. So there's a bunch of non-cellular components that also play a major role. Now, all of these cells and all of these non-cellular components are all working together in something called an inflammatory response. So all of these cells and all of those sorts of things aren't happening in isolation. They're not working in isolation. They're all working together to try and remove and destroy the pathogen. And this coordinated response, where they're all sort of working together, is called the inflammatory response. And you would have absolutely seen this in your body. It's really, really distinctive. So it's when you see, if you scratch yourself, it'll start to turn red. It might swell up a little bit. There might be a bit of pain, a bit of heat. All of that is the inflammatory response working. So the response aims to eliminate the effects of injury, defend against pathogens, and clear out any damage and initiate repair. So because it's innate, it's non-specific. So this is going to happen no matter what. So this is going to happen whether it's a pathogen, an injury, an allergen, a peanut. It's going to happen no matter what. So we're going to go through the three main stages of the inflammatory response. So the first thing that happens is initiation. So this is when the pathogen is originally initiated into the body. So in this example here, we've got this little piece of splinter wood, whatever, they've got a splinter or something, and that splinter had a bunch of bacteria sitting on it. So the first thing that happens is that foreign material and those pathogens are going to enter the skin. So immediately they've already evaded our first line defense of our skin, and they are now inside. So the very first thing that happens is our mast cells, because our mast cells, see these guys here, so they're in connective tissues. They're normally the first ones on the scene. So what they're going to do in response to these foreign antigens is they're going to immediately degranulate and release histamine. And this histamine is going to do many, many things, which we'll explain in a minute. But one of the things it's going to do is it's going to also attract macrophages and phagocytes to the scene, and they're going to release cytokines. So cytokines are going to play a really important role in making sure other white blood cells come to help us out. So that's what the first thing, that's called initiation. So that's those initial reactions that happen to signal to the body that something is wrong. We then have vasodilation. So when you see the redness, the swelling, that's due to this thing called vasodilation. So the histamine that was originally released by those mast cells travels to the blood vessels and dilates them. So dilates means they get wider. So this increases blood flow because they're wider, so more blood can travel through them. And that's really good because if more blood can travel through them, that means more immune cells can get to the site, because we know immune cells travel through the bloodstream. So they can travel there quicker. Histamine also makes them leaky or permeable. So it makes blood vessels more permeable, which means immune cells can easily move through them and to the site. Okay, so we might have some complement proteins coming in, we might have some neutrophils coming in, we might have lots of different immune cells coming in because the histamine has allowed increased blood flow to the area, and you would often see this as that redness, that heat that you feel when you get an injury. So that's vasodilation. Then we have the migration. So now that the blood vessels are ready and they can hold more white blood cells, a number of those innate immune components are going to leave the bloodstream and enter the site. So things like phagocytes, macrophages, neutrophils, they're all going to come. They're going to be guided by the cytokines. They're going to release cytokines. They're going to undergo phagocytosis. We've got our complement proteins that are going to come in, and they're going to opsonize, and they're going to create membrane attack complexes, and they're going to do all the stuff that they do. You might have, all sorts of stuff, mast cells and phagocytes and all of those things that we've learned about are going to start coming to the scene to clear up. And this response is just going to continue. So we're going to have, phagocytosis and clearing and whatever. This is going to continue until the site has been completely cleared of pathogens. So once the body realizes, okay, there's no more foreign antigen here, then they're going to start to repair. And that's where things like platelets and stuff come in, and they're going to start to repair the blood vessels, repair the skin, and it's going to start to scab up, which you've probably seen in your injuries before. So that's the inflammatory response. So all of these secondary immune cells, they're not working in isolation, they're all working together for this specific response, which happens pretty much any time a pathogen enters your body. So a really unique example of the inflammatory response is the allergy response. So normally the inflammation response is just going to sort of just happen in one swoop, but the allergic response is quite unique in the fact that it has two separate things that need to happen. So an allergy is still an inflammation response. It's lots of different secondary immune cells acting together, but it looks a little bit different, and you do need to know how an allergy response actually works. So the first stage of this is called sensitization. So this is your initial exposure to an allergen. So normally you don't get allergic to something immediately. Normally you're exposed to it first, and then after that exposure you start to get the symptoms. So during sensitization, you will be exposed to that allergen. So in this example, it's pollen, which is something that I'm very allergic to. So that allergen is going to enter the bloodstream as it normally would. And this can happen at any stage of your life. I didn't develop allergies until I was an adult. So this can happen at literally any time, which is terrifying. So what's going to happen is that allergen is going to come in, it's going to have its antigens on its surface. And for some reason that we do not know, a B cell is going to recognize that antigen. And a B cell, we haven't learned about that yet. We're going to learn about it as an adaptive immune cell, but it's going to recognize that antigen and it's going to produce antibodies towards it. And these antibodies are going to attach to mast cells. And we know that mast cells are the guys that create histamine, which makes sense because that's the main driver in allergic responses. So in that initial sensitization phase, we've got a reaction to our allergen, but there's no symptoms yet. It's like the setup to the allergy. Then what happens is we're exposed to it again. So it's a secondary exposure, and this is when the reaction happens. Because we've already been exposed to the allergen once, we've already got those antibodies that are formed towards that antigen, and they're already attached to mast cells. So what's going to happen when we get exposed again with the allergen is that there's going to be an immediate response, and that response is going to be mass release of histamine. And histamine is what causes all those awful reactions to a seemingly harmless thing. So an inflammatory response still occurs, so that release of histamine is going to cause that inflammatory response that we learnt about in the last slide, but it's just got that two-step, which is really important to note. So this last slide here is just a summary of everything that we've learnt about. So we've learnt about a variety of cellular and non-cellular components that form the second line innate defense systems. We also learned about the first line, which was, quite simple, those first line defenses that are going to protect us. And we also learned about how all of those components can work together to initiate an inflammatory response to remove a pathogen, because pathogens cause disease. Our bodies don't want that. So we put everything into it that we can to prevent that from happening. Thank you for watching. If you've got any questions, bring them to class and I will see you there.